From the Center for Precision Medicine and Genomics, how genetics can help with the diagnosis of primary ciliary dyskinesia. Diagnostic challenges in primary ciliary dyskinesia. Presented by Claire Keating, MD, Associate Professor of Medicine from the Department of Medicine, Pulmonary.
My plan here is to present to you a case of a patient who came into my office for workup, and then take a back step to do a general overview of this very rare disease of PCD, and then wade into the murky waters of the uncertainty of genetic testing and what the limitations are. Okay, so I'll start with my case. So when he presented to me, he was 30 years old in 2016. So he was a 30-year-old man from India, really no significant birth history, fairly normal infancy. And then when he was a toddler, he was found to have an atrial septal defect and underwent repair, and at the same time was told he had dextrocardia. He says that throughout childhood, he really developed this chronic cough, always kind of a wet cough, sinus infections, and even needed a sinus surgery for polyps in his teenage years. He did deny any history of otitis media or drainage tubes needed. So hospitalized at age 16, this is when he was still in India, and just of note, it's complicated by a bleeding peptic ulcer that required intervention. In the following years, as he entered college, he started having more exacerbations, and he stated up to 6 to 8 times a year he would need oral antibiotics, but was never readmitted. This cough with sputum, which he really described as purulent, sometimes bloody, continued into adulthood. And then in 2016, he decided to move to the US, partly to work at a job at one of the financial banks, and also to pursue a workup. So this workup that I'll present to you was mostly done up in Boston and also at NIH. This is a scout film from his CT scan. And you can see on the view that he does have a right-sided heart. There's clearly a lot of parenchymal changes here. And if you look carefully, probably a left-sided liver as well. And just scanning through his CT. Sorry, I'm not a Mac person. There we go. So upper lobes, fairly well-preserved looking airways and parenchyma. There is a right-sided aortic arch and a left-sided SVC, so consistent with situs inversus. As you see his right middle lobe, there is bronchiectasis, thick and dilated airways, also some mucus plugging and nodules in his lingula as well, and then pretty severe and more advanced bronchiectasis and scarring in his lower lobes, particularly in the right lower lobe. And indeed, reversed on all his organs. All right, so he gave sputum cultures. He grew methicillin-susceptible Staph aureus, Haemophilus influenza, and at the time was negative for any non-tuberculous mycobacterium. He did have a nasal nitric oxide test done, one of the general screening tests that's available at research centers and PCD sites, and it was very low at 11 nanoliters per minute. Below 77 is considered diagnostic, and an average person is certainly 200s and above. Though at the same time he'd gotten married, so he was ready to start a family, but was unable to conceive. So he was doing a parallel fertility workup, and strangely was found to have azoospermia. So you have this patient who, if he didn't have situs inversus, he has situs inversus totalis. So you really suspect he has sinus disease, pulmonary disease, all starting at a young age, that he likely has PCD. But if he didn't have situs inversus, cystic fibrosis would remain on the table as well, because that is the other group and disorder that has low nasal nitric oxide. And the azoospermia is certainly odd. Most patients with PCD would have low sperm counts and immotile sperm. So he went for genetic testing, first just a screen of the CF gene just for delta F508. They actually found a single copy of delta F508, but then on complete sequencing of the CF gene, they didn't find any additional mutations or alterations. Then he had a PCD panel, which is actually fairly broad. It was over 40 to 50 genes that were looked at, but it found only 2 variants of unknown significance in 2 separate PCD genes. And I will explain this a lot more in detail, but for a diagnosis of PCD, you need to have 2 pathogenic or likely pathogenic mutations in the same gene. So again, taking a step back, let's just talk about this rare disease. So I'll really emphasize this is a really heterogeneous disease, predominantly autosomal recessive, though there are some X-linked and autosomal dominant mutations. And what it leads to, if you have any mutation in any one of these PCD genes, is some impact on the cilia, ciliary movement, where they either don't move, they move in a very scattered way, or they're just really blunted and don't do anything, or barely develop. It does occur globally, so it is present in all races and ethnicities across the world, and there's no predilection for gender, for male versus female. The cilia, again, you may remember there's several different kinds of cilia. The focus for sinopulmonary disease and PCD is the motile cilia. So this works as just with general average beat frequency, an innate defense system to keep secretions and mucus and infections fluid and moving through the airways and the ear canals and the sinuses. There's also the embryonic nodal cilia. These are present during embryogenesis, and these are the cilia that dictate organ laterality. And then there's non-motile primary cilia, which I know much less about, but they go with sensory receptors, and they could lead to a lot of these more severe diseases: retinitis pigmentosa, skeletal malformations, and polycystic kidney disease, which maybe Kelsey knows as well. So you can have overlap of motile ciliopathies with some of these other diseases as well, because it all is based on ciliary structure abnormalities. So again, you may remember back from pathology, but this is the ciliary structure, 9+2, 9 doublets around a central pair. And among these, this whole arrangement are inner dyning arms and outer dyning arms. These are the large motor protein complexes that help ciliary movement. And then there's Nexon links and radial spokes. Is this seeming familiar? So those help the ciliary structural supports and sort of keep it upright. And these are just various pictures of electron microscopy, which is a very specific and highly specialized preparation. So when one is sending patients' cells off for electron microscopy, you really need to have a pathologist who knows how to prepare the cells and how to analyze the cells, and also proper collection of the cells, generally from nasal brushings or nasal biopsies. So just quickly, panel B is a normal structure. You might have to trust me on this one, but panel C is outer dining arms missing. Panel E is inner dining arms missing. I'm not sure what's up with D, but the F and G are clearly abnormal. But again, another pitfall, and this is not even getting into genetics, 30% of patients with PCD will have normal ciliary structure, even with proper processing. So clinical manifestations, it all starts in really infancy and childhood. I didn't write it down, but 80% of patients will have neonatal respiratory distress. Majority of children just have that chronic wet cough throughout childhood, often interpreted as allergies and asthma. They start to develop true lower respiratory tract infections by preschool, and almost 100% of patients will have bronchiectasis by the time they reach adulthood, predominantly the lower lobe and middle lobes, as opposed to cystic fibrosis, which starts in the upper lobes. Due to recurrent otitis media in majority of kids, they will need drainage tubes. They will have chronic perforations, chronic scarring. It can lead to speech delay in the little ones due to conductive hearing loss, just from chronic effusions and chronic purulence in their ear canals. And as mentioned, they can have laterality defects, pectus excavatum, scoliosis, and congenital heart disease. Quickly touching on that, this is just a review of patients in North America in the PCD registry. Of 400 patients, a little over 400 patients that were looked at, if you scroll over here, so a little less than half will have situs inversus totalis, and that's what we generally believe. About 50% of PCD will have complete reversal of organs, and a large number will have situs solitis, everything on the right side. But there is a subset, about 12%, at least according to this review, have some amount of heterotaxy, or otherwise called situs ambiguous. A lot of this is congenital heart disease. So either a very simple, like our patient had just an ASD, but about 2 to 3% will have actually quite complex congenital heart disease requiring surgeries. Some can end up with Eisenmenger's. So this can really complicate managing a patient because they will be hypoxemic for other reasons, chronically short of breath, and you have to partner with their cardiologist. And then the rest, which are very few, can have malrotation of the gut, polysplenism. One thing's on the right side, one thing's on the wrong side. So if you have a patient who does have sinopulmonary issues and some amount of heterotaxy, this should be at the top, should be on your radar. So diagnostics, I mentioned electron microscopy. There's also video microscopy analysis, actually looking at beat frequency, obviously available at very few highly specialized centers. The nasal nitric oxide, not widely available, but is mandatory for any center that is accredited for PCD. So we do have one housed in the Children's Hospital, and then genetic testing. All right, so what is the prevalence? So going back to the papers from the 1950s and 70s and kind of pass forward, it was estimated to be really, really rare, about 1 up to 1 in 20,000 patients. And this was mostly just based on X-ray data, those with laterality defects, provider surveys. This predated genetic testing. The PCD Foundation, which is a registry of all of North America, as of their last estimation, only about 1 or 2,000 patients are accurately diagnosed, but they believe probably 1 in 25,000 people are walking around with PCD in North America. And this is due to a multitude of factors that I've listed here. All right, so this, I just want to quickly highlight 2 interesting studies. This was published 2 years ago in the Lancet, really trying to get how common or uncommon is this disease. So these researchers looked at 2 different databases. They took data from Invitae, which is a commercial genetic testing company, and they looked at just 25 genes from their database with a known pathogenic or likely pathogenic. And they admit they've left out about 30% of other genes because they were poorly defined or characterized or weren't discovered at the time of their research. Then they cross-checked it with Genomad, which is the genome aggregation database, is that right? Which is it's a collection of multiple sequencing data sets of up to 14,000 individuals. And they looked for these pathogenic mutations, 2 in 1 gene in this large data set. And they found that the prevalence of PCD worldwide, again, these data sets were global, is actually down about 1 in 7,500. So we believe it's actually much more common than we believe. Also this is also, they intentionally wanted to include a wide range of areas of the world, ethnicities, and backgrounds. And there are certainly certain mutations that might be more common in one background or another. Clearly, there's some that are most common. DNA H11, for example, is the purple, most common in sort of across all patients, but you might find certain tendencies to certain areas of the world or backgrounds. And I didn't put it here, but interestingly, in this study, at least, they found that it was most common in African or African-American populations. And in their database, it was 1 in 200 were diagnosed with PCD in this analysis. Also, kind of backing this up is also an interesting project that published a few years ago, the UK 100,000 Genomes Project, where their goal with this, what started about 10 years ago, was to get whole genome or whole exome or whole genome sequencing of over 100,000 patients with either rare disease or cancers, either them or their family members. So they pulled from this data set those with severe lung disease, specifically FEV bronchiectasis, FEV1 less than 30% predicted, or less than 50 years old with multi-lobe bronchiectasis and a suspected inherited disease. And importantly, they excluded people that they had a high suspicion of PCD because they were recruited for a different study through the same project. And when they came down to the remaining patients of 142, they found that 12% of them screened in positive as having 2 mutations in a single gene and diagnosed with PCD. Again, that this is likely much more prevalent disease than we believe. So when you look at the American Thoracic Society guidelines, genetics is part of every pathway in the workup. Again, I know it's very fine print, but what it says here in the gray is, can you get a nasal nitric oxide or not? It's an easy, non-expensive test. Actually, you can't charge for it, so it's actually free. That if you can do it, you should get the nasal nitric oxide, but you should always go to confirmatory testing. So yes, they have a low nasal NO, but please get genetic testing or electron microscopy. If it's a normal nasal NO, there are patients who have PCD with normal nasal nitric oxide. You should get your genetic testing if you have a high suspicion. And on this other pathway, you have no access to nasal nitric oxide, you should get genetic testing. So it really is part of every workup. So how many genes are there that we're hunting for when we send a patient for workup? As of this year, there's 53 genes that have been identified, and this kind of continues to grow. As you can see, it's a wide range of mutations, and within each gene, there's a varying amount of coding exons too, between 2 or 3 on the left hand up to 85. Whoops. So this shows, and where are these? These are all over the genome. They cross over 18 chromosomes, as opposed to like a monogenic disease like cystic fibrosis, single gene, chromosome 7, it's there, it's not the mutation, they're not there, they're well classified. This is quite a large spread. And I think the takeaway from the previous slide and this one is that there are so many genes that contribute to ciliary formation and structure and function, and any mistake in any one of these genes can cause downstream effects of varying phenotypes. Or the genetic counselor who sent me these slides, just said basically there's so many ways to break the cilia, and that's what makes it such a complex disease. So is there a correlation between what gene you have and how severe a patient is? It's not so straightforward, but this left column are common mutations. DNAH5, DNH11 is in the second column. But this first column, these have abnormal electron microscopy. And within this range, these are mostly the outer dining arm and inner dining arm defects. These patients can be more severe, especially the CCDC39s and CCDC40s. And so some patients will go on to lung transplant, and it is more of these more severe downstream effects. This middle column, while these are, some of them are common, these will have the normal electron microscopy. And then the last one is much, much less common ones, including the X-linked and the autosomal dominant. Okay, so our patient really wanted to get an answer. He's very was on focus to like, do I have PCD? Do I not have PCD? At the NIH, they did send him for whole exome sequencing, and they did find 2 pathogenic mutations in the C11ORF70 gene, which is on that list, but was not available on the commercial panel. So, and I have a few more slides and then I'll wrap up. But I think what this highlights is that when you do a genetic test, it depends what lab are you using, what is their panel, how inclusive is that panel, and even if they find mutations, what is their expertise and database and knowledge to understand whether or not those genetic variations are truly pathogenic or not. And then it gets even more complicated if you send someone for whole exome sequencing or even whole genome sequencing, because then you need an analyst who really knows which are the PCD genes that could be culprit, and how do you interpret if those mutations have a significant downstream effect. And I know there's a very complex algorithm in many databases that they use in terms of gene preservation over time and other ways they can see, would this mutation actually cause a clinical defect? So takeaways, there are many genes associated with ciliary dyskinesia. Not all genes are on test panels. This can differ from lab to lab. Invitae is the one we most commonly use, but even Invitae misses the Haydn gene, which apparently is more common than we think, and you have to go to Blueprint Genetics for that one. So it's really a decision-making, will insurance cover it? And again, will the analysis be accurate? Not all genes are known. So with the panel of 53 genes, about 70% of patients can be identified, but that leaves it open for 30% who have unknown causative genes. Whole exome sequencing can help and identify a gene, but you have to have enough information to interpret. And then there's the whole area of what about non-coding regions? So these include, they mostly just do the exon regions and maybe a little bit out into a handful of base pairs beyond, but there are known mutations in deep intronic regions. There are known mutations in promoter regions, and these are not interrogated, certainly by commercial labs. And you have to kind of know what you're looking for. So if you're really going down that route, you often end up in some research geneticists' hands to really dive deep. So finally, why do we care about a diagnosis? There are really no PCD-specific medications out there. We just treat them like most of our other non-CF bronchiectasis patients. But number one, we do know that early intervention across all forms of bronchiectasis does limit or at least slow disease progression. It gives patients access to multidisciplinary care. So not just a pulmonologist who has expertise in bronchiectasis, but a physical therapist, a dietitian if that's necessary, a social worker. Having a coordinated center that can refer to specialists as needed. So you really need a good team. You need good ENTs, including the rhinologists and those who manage the otitis issues, early referral to fertility centers for the men, but even women have reduced fertility. And is your patient really being cared for if there is a cardiac issue that needs to be closely monitored? And then finally, identifying these patients, if you don't know who they are or where they are, how are you going to find people for clinical trials? And which I'm not going to go through today, but there have been randomized clinical trials in PCD. And one kind of common theme across them is that they never meet enrollment goals. They do their best, but it's hard to find, especially adults, which are underdiagnosed even more than the kids, to actually participate in these trials. In addition to typing them, finding what the needs are, including genetic therapies. So it really is important to find these patients and get them connected with centers and then into the national database.
This was brought to you by the Center for Precision Medicine and Genomics at Columbia. This has been an internal medicine genetics case study.